and enhance the grain yield by 7 to 11%. Now, when we say saving of 30 kilogram phosphorus, that means you have 60 kg in phosphorus per fertilizer, curtail the use of chemical fertilizers, and you can improve the productivity of the soil also. Now, besides the solubilizing organism, there is another category which we say as mobilizers. Mobilizers are the ones which mobilize phosphorus from the soil and it gives directly to the plant. So there is a fungi, there's a group of fungi which we'll call as arbuscular mycorrhiza fungi. Now, this arbuscular mycorrhiza fungi, they are obligate symbionts. That means they can survive only in the roots of the plants. You cannot culture them in the lab, just like any other bacterial inoculant you can grow in the lab on a particular medium. But this arbuscular mycorrhiza fungi cannot be grown on the medium, and therefore it has to be maintained only on the roots of the crop plants. So every time you have to maintain a crop on which these mycorrhiza can survive and can multiply. So mostly grasses like Sancrum ciliaris and even the maize crop is used for multiplication of these arbuscular mycorrhiza fungi. Now, these fungi, the importance is that it forms a connection between the deeper layers of the soils and within the roots. That means the mycelia is present in the soil also, it is present in the roots also. So there is a direct connection between the soil and the roots. And this mycelia can mobilize the phosphorus from the soil into the roots. So they are helping the plants by uptake of phosphorus, and later on it was found that besides phosphorus, many other trace elements like zinc, iron, copper, cobalt, magnesium, and molybdenum, these can also be mobilized by these arbuscular mycorrhizal fungi. And not only the mobilization of microorganisms, mycorrhiza has been found to reduce the effect of salinity, and it also checks soil erosion and reduce the loss caused by the nematodes. So it is a very important inoculant, and whenever the crops are being inoculated with AM fungi, it has always given a very good response. Now, as I said, mycorrhiza, AM mycorrhiza, it is an obligate symbiont. That is, it cannot be cultured on the medium, but it can be cultured only on the plant. Later on in 90s, another organism was identified, which is called as the Piriforma spora indica. This is again an endophytic fungus, but the advantage is it can be cultured. It has functions almost similar to AM fungi. It can also mobilize phosphorus. It can also mobilize other micronutrients. And the advantage is that it can be cultured on the medium. So this Piriforma spora indica, it also provides nutrition to the plant, and it also has a suppressive effect on many of the fungal pathogens, like the slide shows the suppression of the powdery mildew disease. So these are the different organisms which we started working on, say, from 1970s to 2000. And these are the organisms which became popular. Now, when I started, I showed you a small bar, which is a red bar, which I said that I'll be discussing later on. So in 2014 and 15, there is some changes that took place in the biofertilizer technology. And these important changes were the development of the liquid inoculants. Now, these liquid inoculants, they have an extended shelf life. Most of the carrier-based inoculants, the shelf life is maybe for 3 to 6 months. When I say shelf life, to make you understand, shelf life means that the colony forming units of the bacteria which are present in the formulation, it should maintain about 10 to the power 5 into 10 to the power 7 cells per gram of the carrier or per ml of the liquid inoculant. Now, most of the time when we are using carrier-based inoculant, the population count goes down, and therefore such type of inoculants cannot be used in the field. Now, with the liquid inoculants being developed, the advantage was it has a shelf life of about 12 months. It can be kept for a longer period of time, and therefore it can be used even after a year in the field. The second change which took place after 2014 was the number of bioinoculants increased. As I said, initially we were working only on 5 organisms like Azotobacter, Azoospermum, Rhizobium, PSB, and blue-green algae. But now, after 2014, many new categories of biofertilizers, they also came into being, and the production of these inoculants started. And the important ones were the zinc solubilizing bacteria, K solubilizing bacteria, formulations of Pseudomonas fluorescens, Bacillus, and many other PGPRs. So this was another change in the bioinoculant industry which took place after 2014. Then the third major change which took place after April 2014 was, this is just to show you how the Gazette of India, it describes, whenever a new inoculant can be prepared by the manufacturers, all these things are notified by the government. Now, for the first time in 2014, the government gave permission for the development of consortium. Consortium means where 2 or 3 microorganisms can be mixed together. So they gave specifications for a consortium of nitrogen fixing, E solubilizing, and K solubilizing bacteria. That means all the 3 major nutrients, nitrogen, phosphorus, and potassium, they can be provided by a single formulation. So this was a major change which came in 2014. And afterwards, the consortium also was manufactured in the country. Then the fourth change which took place was that the increase in the production. If you see the figures of 2008 and 9, it was only 25,000 metric tons, which increased to 80,000 metric tons in 2014-15. The reason was that the government of India started promoting the production of biofertilizers in the country through various programs under National Mission on Sustainable Agriculture, Mission for Integrated Development of Horticulture, Rashtriya Krishi Vikas Yojana, Network Project on Organic Farming. So, because of the initiatives taken by the government of India, the production of these biofertilizers enhanced, and many of the ICR institutes and many of the state agricultural universities, including TNAU, Coimbatore, US Bangalore, US Harvard, they also started producing these biofertilizers on a small scale. So these are some changes which took place, and thereafter new biofertilizers, they came into being. As I said, these liquid bioinoculants, they are a definite improvement over the carrier-based inoculant because they can be stored at elevated temperature up to 45 degrees centigrade. They have a shelf life of more than 2 years also, as compared to 6 months of carrier-based formulation. And they do not show any loss of the plant growth promoting activities in the formulation. And the microbial load in these liquid bioinoculants can be very high, up to 10 to the power 10 cells per ml. So these liquid bioinoculants are now becoming popular because they are convenient to use as compared to the carrier-based inoculants, which were more cumbersome as compared to the liquid inoculants. So the new biofertilizers which we started producing now is a potash solubilizing bacteria. Just like the phosphorus solubilizing bacteria, these K solubilizing bacteria, they also solubilize potash, again by production of organic acids, and they also provide potash to the crop plants. So amount of K solubilized or the amount of potash which these organisms can supplement is about 10 to 15 kilogram per hectare. So these formulations are now being used to improve the growth of many different crops. So one example was in case of sorghum, grown in Andhra Pradesh, and it saved about 25% of the K fertilizer. Similarly, zinc solubilizing bacteria, again, this type of inoculants were not in use before 2014, and after 14, these zinc solubilizing bacteria were used for production of biofertilizers, and they can help to increase the seed yield as well as the zinc uptake by the seeds. Now comes very important formulation, which we call as bio-NPK liquid microbial formulation. It has been developed at our bureau, and as I said, the government gave a green signal that the consortium can be prepared. So we have prepared a unique formulation which has a nitrogen fixing organism, a P solubilizing and a K solubilizing organism. Now when we apply this type of a consortium, it helps to augment about 25 to 30 kilogram nitrogen, 20 to 25 kilogram phosphorus, and 10 to 15 kilogram of potash per hectare. So you can now see the effectiveness of a single formulation. You can save all the three major nutrients to a greater effect, and can reduce the cost of cultivation by reducing the input cost. So this type of a bio-NPK formulation, it has been tested for many different crops like rice, wheat, maize, groundnut, fodder oat, and berseem. Then besides these grain crops, it has been also used to improve the growth of many of the horticultural crops like the citrus, and also has been tested in turmeric, ginger, some of the flower crops like marigold and gladiolus. So such formulations or such biofertilizer, when we are using, they help to cut down the cost of the chemical fertilizers or the use of the chemical fertilizers. And they also improve the fertility of the soils by providing all the three major nutrients. Then nowadays, these microbial inoculants, besides providing only the major nutrients, they are being used for biofortification. I'm not sure how many of you are aware about that there's a deficiency of iron and zinc in many of the human beings, and this is what we call as the hidden hunger. So in India, many people suffer from these deficiency, and the only way is to fortify the grain crops so that when we are consuming these grain crops, we can add iron and zinc to our body, and they can supplement both the micronutrients to the human beings. So we have been testing different organisms, and some of the organisms which can enhance iron concentration in the wheat grains are Arthobacter and Enterococcus. Similarly, we have identified organisms which can enhance the zinc concentration in the wheat grains. So if we are using such type of fortified grains, it can help to remove the hidden hunger, which is basically because of the deficiency of iron and zinc. Now, these microbial formulations, they are also used to alleviate the stress, particularly the drought stress and the salinity stress. So we are now growing our crops using Archaea and bio-NPK without giving any irrigation. And this is one example of mustard. Similarly, we have done experiments with wheat where without any irrigation, at least we were able to get 22% of the yield as normally obtained from the irrigated crop. So although there is a considerable reduction in the yield, but in places where moisture stress is a problem, inoculation of these inoculants can benefit the crop plants. So coming to the end, I would like to say that these biofertilizers which I have talked about, they are not the replacement of chemical fertilizers, but can supplement the plant nutrient requirement. Hence, judicious planning of using biofertilizers in combination with inorganic fertilizers will definitely have a long way in saving our fossil fuels and protecting the environment. So thank you very much for your patient hearing, and thanks the organizers for giving me this opportunity. Thank you all.
Your thoughts have enlightened our minds and have shown us a new path. My gratitude to all the speakers for gracing the occasion and sharing their opinions today. Thank you, sir. Thank you, everybody. Thank you once again.
Thank you.
Thank you, sir.
Thank you. Thank you. बोल दो जनरेटर बंद करवा देगा जनरेटर हो गया काम मेरा हाँ तो इसलिए चलवाया था बीच में रुक ना जाए प्रेजेंटेशन दे रहा था ना अभी